Before we begin, I'd like to make a brief land acknowledgement. McGill University is a land which has long served as a site of meeting and exchange amongst Indigenous peoples. We acknowledge and thank the diverse Indigenous peoples whose presence marks this territory on which we now gather. Just a reminder to please leave your questions till the end. Please enter them in the chat. And as well, please enter a brief survey that will be made available to you after the presentation. And finally, I have the honor of presenting our speaker today. And this is a special round. This is our Lucian Award recipient for 2026, Dr. Ziad Mallat. Is it's the 2025th Lucian Award, no, 2026 Lucian Award. We're kind of in both in circulatory disease. Professor Mallat works at the University of Cambridge. He is the recipient of numerous other awards, including the Jean-Paul Binet Prize from the Fondation pour la Recherche Médicale, the Jean Hamburger Prize from the City of Paris, and multiple honors from the French Academy of Sciences and the American Heart Association. He is among the world's leading clinician scientists in the area of atherosclerosis and aneurysm. His early work was groundbreaking as he identified an important atheroprotective role of regulatory T cells and associated anti-inflammatory cytokines. The work was further advanced when he identified selective pathogenic and protective roles for defined B cells in innate lymphoid cell subsets in atherosclerosis and cardiac remodeling following ischemic injury. More recent work demonstrated that subpopulation of natural and or inducible regulatory T cells can inhibit atherosclerotic lesion developments and inflammation. Through this work, he aims to develop novel immune-modulatory and vaccination-based strategies to promote protective immune responses in cardiovascular disease. Dr. Mallat, a real pleasure to have you here today. Welcome.
Yes, thank you very much for this kind of introduction. I think my talk here will be going to present to you what we know about the role of the inflammation and the immune system in general in atherosclerotic cardiovascular disease, and giving you really what I think about the published data and where we are going from this. So this is the general contents. What is, the relationship between the most important atherosclerotic cardiovascular risk factors, LDL cholesterol, and inflammation, going through the main innate immune mechanisms of cardiovascular disease and their related treatments and what I think about it, and then talking about main adaptive immune responses and how to target them. So, as I said, LDL cholesterol is one of the most important risk factor for atherosclerosis. Jan Boyen has worked a lot on the atherogenicity of LDL cholesterol particles, and we know now that the concentration of LDL cholesterol in the blood is important because it determines also the time needed for them to enter the artery wall when there is alteration in endothelial permeability. And in fact, in order for the LDL cholesterol particles, in order to start the atherogenic process, they need to be modified. And there are a lot of different kind of modifications, oxidative and non-oxidative modifications. And an important step is their trapping in the extracellular matrix beneath the endothelial layer. And once they become modified, then they expose epitopes that are recognized by the immune system and activates endothelial cells and vascular smooth muscle cells to express chemokines and adhesion molecules, and this goes to induce inflammation. And in fact, we know from very simple studies, I think that the most important studies are the simplest one. They are really come from great thoughts and thinking. This is an experiment where Jan Boyen's group have overexpressed the human ApoB100 in mice. And in fact, they deleted in these mice the proteoglycan binding domain of ApoB100 to limit its trapping in the extracellular matrix. And you can see that there is a correlation, direct correlation between cholesterol and lesion size in the wild type mice. And for the same level of cholesterol, you have a huge reduction in atherosclerosis in the mice where LDL cannot bind and be trapped in the extracellular matrix. So this is a very important step in atherogenesis, and this is where increasing residence time increases also the modification of these LDL to become inflammatory. So they become inflammatory and they attract monocytes and T cells into the lesions. And about monocytes, we know also from very good experiments that monocytes are essential for lesion development in mice. So you can have high levels of LDL cholesterol. If you don't have inflammation, in particular, if you don't have monocytes, you cannot develop atherosclerosis, despite the fact that these mice have high levels of atherosclerosis. So Jonathan Smith had published this paper where he looked at mice that are deficient for MCSF, monocyte colony stimulating factors. In these mice, there are no or very low levels of monocytes and macrophages. And despite the fact that they had very high levels of LDL cholesterol, they had no atherosclerosis. So you can have LDL cholesterol elevated, trapped, induces, and it needs to induce inflammation in order to go and make atherosclerosis. So let's stay with innate inflammation. And we know, from human studies, this is an example of human studies by Paul Ritker and others, that systemic markers of inflammation, and here high sensitivity C-reactive protein, are associated with the risk of atherosclerotic cardiovascular disease. And this association, there is an interaction, in fact, between high levels of systemic inflammation as measured by high sensitivity CRP and total cholesterol in promoting increased cardiovascular risk. And you know that from new studies from the same group, the association of, so there is an interaction between inflammation, cholesterol, and other lipids, and this study shows you the interaction between inflammation as measured by high sensitivity CRP levels and LDL cholesterols and lipoprotein little a in inducing risk. The more you have of these biomarkers elevated in the blood, the higher the risk of cardiovascular events. And in fact, it's not just an association. We know from genetic association studies, so potentially causal mechanisms of atherosclerosis, that inflammatory pathways are also potentially causally related to atherosclerosis. And you all guys know about the association of this genetic variant in the IL-6 receptor and the susceptibility to atherosclerotic cardiovascular disease. There are other potentially inflammatory mediators, interleukin-5, chemokines like CXCL12, etc. So there is also, these genome-wide association studies have also shown that inflammatory pathways may be causally associated with the risk of coronary artery disease. And not only, genetic variation, but also somatic, newly acquired somatic mutations can also predispose to increased risk of coronary artery disease. This is the example of mutations in genes that are involved mostly in epigenetic variation and increased risk to hematologic cancer, DNMT3A, TET2, etc., that they have been shown by several groups now that the presence of the increased accumulation of these mutations over time, their accumulation increases with age, and the presence of these somatic mutations is associated with increased risk of cardiovascular disease. And in fact, one of the most important association of somatic mutations in terms of predisposing to clonal hematopoiesis and cardiovascular risk is the TET2 mutations in the TET2 gene. And these mutations are associated with increased inflammatory potential. And in particular, this work from Ken Walsh and others have shown that TET2 mutations are associated with increased activation of the NLRP3 inflammasome pathway and increased production of IL-1 beta. And as you can see here, TET2 mutations increase atherosclerosis but this increased level of atherosclerosis can be limited by inhibitors of the NLRP3 pathway. And in fact, these studies and others that have shown that NLRP3 inflammasome pathway is involved in atherosclerosis and leading to, this pathway leading to the production of IL-1 beta in particular, have led people and investigators to look at what is the impact on cardiovascular events of blockade of IL-1 beta derived signaling pathways. And you know all about this CANTOS trial that tested different doses of canakinumab, which is a monoclonal antibody against IL-1 beta, in patients with stable coronary artery disease, but at increased risk of cardiovascular events because they have high CRP levels. And you can see here that in these studies, people have shown that canakinumab treatment reduces drastically high sensitivity CRP levels, indicating that there's a reduction in systemic inflammation without changing LDL cholesterol levels. And this was associated with a significant, although relatively modest, I mean, we can discuss about this, 15% reduction in the risk of cardiovascular events in this population. Now, if you look at this, and in fact, there are new studies showing that, I told you that CHIP mutations and the TET2 mutations in particular are associated with increased NLRP3 inflammasome and increased production of IL-1 beta. And a retrospective analysis of data from the CANTOS trial have shown that this reduction of cardiovascular events that they have seen is mostly, in fact, seen in the patients with CHIP mutations, in fact, with TET2 mutations. You can see that the TET2 mutations, patients with TET2 mutations derive more benefit and reduction in cardiovascular risk in comparison with patients with non-TET2 mutations. Now, this is a very retrospective analysis, and it's just exploratory analysis, but it tells you of the interaction between TET2 IL-1 beta signaling pathways and cardiovascular disease. Now, this therapy has not been approved for treatment in these patients because of, as you know, I mean, people can say that this was not the reason, but I really believe that this was the reason of non-approval of this therapy for treatment of patients with cardiovascular disease is the increased risk of fatal infections. And again, I think that this is even more important in patients who are older, and we are treating more and more older patients who are highly susceptible to infections. And you don't want to have a therapy that reduces cardiovascular risk, but in the same time can increase the risk of fatal infections. So there are other therapies that have been trialed in order to, and have been, presented as they are targeting the same pathway of NLRP3 IL-1 beta. And I will show you some examples and go through the interpretation of these data. Colchicine in patients with chronic coronary artery disease, the LODOCO 2 trial. So this is a randomization to placebo or low-dose colchicine in these patients with chronic coronary artery disease. As you can see, the primary endpoint, which includes here not only MACE, but also a revascularization events. There was a significant reduction in cardiovascular events in these populations. However, and this is where I'm giving you my own interpretation of the data, because I think that some people, to some extent, we all overlook some important data to be interpreted. So, in this New England Journal of Medicine paper, there was, the study was done in two countries, Australia and Netherlands. And I'm wondering how many of you know about this. In the paper, they said, when examined according to country, the effect of colchicine as compared with placebo was directionally consistent, but appeared to be quantitatively larger in Australia and Netherlands, figure supplementary 2. So this is figure supplementary 2, and these are the results. So if you don't go to supplementary figure 2 and look at yourself at the results, okay, so the trial was positive in Australia, was negative in the Netherlands. I mean, you can still say that there is the direction is the same, but in fact, the confidence interval is quite large. And in fact, two-thirds of the patients were recruited in the Netherlands and only one-third in Australia. So, I mean, there's a kind of different interpretations. And you all know about, colchicine is very well known in Montreal by the work of Jean-Claude Tardif, who did many studies on this. And this is one example of low-dose colchicine in patients after myocardial infarction, and reported a significant reduction in cardiovascular events here. But in fact, if you look at the data, most of the reduction in cardiovascular events here were in urgent hospitalization for angina leading to revascularization, rather than a reduction in myocardial infarction or death from cardiovascular disease, etc., and also a significant reduction in stroke events. Now, there is this new study in patients with acute myocardial infarction, which is the CLEAR Synergy trial. In patients with acute myocardial infarction, of course, there's some difference between this study and the previous study in terms that this study included also ST elevation, a lot of ST elevation, myocardial infarction, but there is absolutely no impact of colchicine on cardiovascular events in this study. There was some suggestion that because of the COVID-19 events, etc., I mean, you can say what you want about studies. Nevertheless, this was quite negative results. And if you look at low-dose colchicine in other trials, looking at cardiovascular events, for example, this is the CONVINCE trial of long-term colchicine in the prevention of vascular recurrent events in non-cardioembolic stroke. There is, if you look at comparison with usual care alone, because this was an open-label trial, you still have, some higher reduction, bigger reduction in CRP levels in colchicine in comparison to non-colchicine groups, which is interesting. However, there was no significant impact on the cumulative incidence of cardiovascular events in this trial. I'm not advancing this anymore. Oh, sorry. So now there is a recent meta-analysis on all these trials, combining all the colchicine trials together, including, the trials in stable coronary artery disease and unstable coronary artery disease. And the agreement is that, this meta-analysis shows that there is a reduction in MACE, but, the relative risk of reduction is much lower than that has been published, at least before in individual studies, around 12% reduction in MACE with the increased risk of cardiointestinal events. Now, if you look at these studies, these are the different studies that are included in the meta-analysis. You can see that the biggest ones, CLEAR, CHANCE-3, are quite negative. And in fact, when you look at CONVINCE and LODOCO here, these were, remember, open label trial, not a blinded trial. And in fact, if you look back at what was the reason, to test colchicine in patients with cardiovascular disease, the first reason was that colchicine was an inhibitor, a known inhibitor of the NLRP3 inflammasome pathway, and this might reduce IL-1 beta production. So this is the first study that has been published showing that microtubule-driven, because colchicine, its impact on microtubule polymerization. And this is the study published in Nature Immunology that showed the impact of colchicine on NLRP3 inflammasome pathway, showing that increasing doses reduce IL-1 beta production and NLRP3 activation. However, when you look at the doses, the concentration of colchicine used to inhibit NLRP3 inflammasome, they are in the micromolar range. This is 5 to 15 micromolar. This is 100 micromolars used in order to inhibit NLRP3 inflammasome. Now look at the concentrations that you can achieve in vivo with colchicine in human beings in the cells, not only in the circulation in the plasma, but also we know that colchicine is is enriched and concentrated intracellularly in some immune cells like polymorphonuclear cells or mononuclear cells. But look at the concentrations, and these are nanomolars, not micromolars, okay? So even when you highly concentrate your product, the colchicine, in cells, it's still in the, mostly, you know, in the tens and hundreds nanomolars. We needed, you know, people needed tens and hundreds of micromolars of colchicine in order to inhibit NLRP3 inflammasome. So I can tell you from this interpretation that I don't believe that colchicine, low-dose colchicine in vivo is acting through inhibition of NLRP3 inflammasome. I mean, colchicine have other different effects. You know, it's a very good inhibitors of neutrophil activation, which could be a way by which maybe colchicine is reducing cardiovascular events in some settings. But I think that certainly not through the NLRP3 inflammasome pathway. In fact, the NLRP3 inflammasome pathway, again, there are suggestion that besides IL-1 beta, you know, the NLRP3 inflammasome activation goes to activation of IL-1 beta, production of active IL-1 beta, but also active IL-18. And this is work from Paul Ridker suggesting that plasma levels of interleukin-18 are also associated with cardiovascular events and an interaction with interleukin-6 and others. So there is the suggestion that in addition to IL-1 beta inhibition, or potentially in replacement of IL-1 beta inhibition, maybe IL-18 inhibition could be interesting. In fact, my group was the first group to show that IL-18 pathway is pro-atherogenic in mice and translation potential in humans. But to be honest, myself, I don't think that this is a very important and promising pathways, because it's also involved in fighting against infections, and there is no clear genetic evidence that these pathways can be important in human beings. And in fact, if you look at the NLRP3 pathways, a lot of people are now, you know, a lot of pharma are developing NLRP3 inhibitors, and some of them are being suggested to be used in setting of cardiovascular disease, because with the NLRP3 inhibitors, you can inhibit the production, as I said, of IL-1 and IL-18 in the same setting, and potentially limit cardiovascular disease. But look, if you have some side effects with IL-1 inhibition alone, why wouldn't you have the same side effects with inhibition of both IL-1 and IL-18 in the same time? And in fact, to be even more, you know, challenging, having challenging data, I haven't seen any publication in the literature indicating that the NLRP3 inflammasome pathway is causally linked to cardiovascular disease in terms of human atherosclerotic cardiovascular disease. So if you look at the GWAS studies, currently there is no indication that this pathway is causally related to cardiovascular disease, but probably additional investigations will come up. Now, the other pathway that I showed you very quickly that is causally, you know, identified by GWAS studies to be related to coronary artery disease is the IL-6 pathway. And you know that there are now ongoing studies targeting IL-6 in patients with cardiovascular disease, and targeting IL-6 reduces significantly, in fact, it suppresses high sensitivity CRP levels in the blood, because again, when you measure hsCRP in the blood in patients, in humans, it is indicative exclusively of IL-6 signaling. You know, there are patients with inactivating mutations in the IL-6 signaling pathway in humans. These patients have zero CRP levels in the blood, despite the fact that they have other types of inflammation. So when you hear about high systemic inflammation by measuring CRP, it is exclusively related to the IL-6 pathway. And you can see that when you inhibit IL-6 here, you have 92% reduction in CRP. So it confirms So we will be waiting until the end of this year, probably, about the outcome of these trials. And there are other anti-IL-6 antibodies that are being tested, and all they act well in reducing CRP. In fact, some of them, we should still be careful, because some of them may also increase the risk of infections, even in small studies. But we will know whether this is, I mean, you expect that this pathway is important, but I mean, we can discuss about it. However, remember that the genetic variant is in the IL-6 receptor, and the genetic variant is associated, of course, with a reduction in CRP levels, which indicates that the membrane signaling pathway of IL-6 is reduced. However, the same variant is associated with high soluble IL-6 receptor and high IL-6 levels in the blood, which might have different impact on trans signaling. So these studies that are directly inhibiting IL-6 are not reproducing what the genetic variant is doing in vivo. So we will see what will happen. And the other things that I would like very quickly to mention is that when you think about genetic variation and relationship with cardiovascular risk in humans, the genetic variant is reflecting a lifetime exposure, right? From development in utero to the adult stage. And when you have a variant in the IL-6 receptor that is potentially acting during the whole lifespan, in fact, my point here is that you don't know whether this variant, its impact on atherosclerosis was mostly during development, during early lesion development, or whether it is important for the destabilization of stable plaque to unstable plaques late during plaque development, okay? And we are treating patients now with anti-IL-6 inhibitors, mostly at the very advanced stage. Imagine that this variant was acting on atherosclerosis mostly during development and is not more active during the advanced stage, you will not see any impact. And I'm saying this, I hope really, I all the time say, I hope that these trials of our anti-IL-6 will be positive, because the problem is they are negative. People will say, okay, there is no role of inflammation in cardiovascular disease. And in fact, people are already saying this when, after the negative results of the colchicine trial in Clear Synergy. I mean, my colleague, Kevin Williams, who is an expert in lipidology and not in inflammation, published these key points, these viewpoints about the fact that, if colchicine is not important in reducing cardiovascular events, then inflammation is not important. And in fact, I discussed with Kevin several times and tell, okay, Kevin, you don't treat LDL cholesterol, all lipids the same. You don't treat LDL cholesterol like HDL cholesterol, like lipoprotein little a, etc. And then you cannot treat inflammation, all inflammatory pathways the same. So there are differences between different inflammatory pathways, and maybe we should look at more in detail at what the immune system is doing. And I will not go into the details here, but you can see just from this slide of review that there are a lot of types of immune cells involved in atherosclerosis, a lot of type of cytokines, etc. And then it is important to understand which ones are the most important and at what stage of lesion development. And the rest of my talk will focusing about the role of lymphocytes and how we can harness this knowledge for treatment of cardiovascular disease. So as I've shown you before, that in the absence of monocytes, there is no atherosclerosis. We know also from experimental studies that if you have no lymphocytes, there is a drastic reduction in the development of atherosclerotic lesions. However, lymphocytes come also with different flavors, and not all lymphocytes are the same. This is the summary of what we know now about the adaptive T-cell immune responses in atherosclerosis. These oxidative specific epitopes can be presented by loaded on MHC class 1 and class 2 molecules to activate CD4 and CD8 T cells. And we know that T helper cells of type 1 producing interferon gamma promotes atherosclerosis. And we also know that this reaction is counter-regulated by what we call regulatory T cells that produce anti-inflammatory mediators like IL-10 and TGF-β and counter-regulate and are important in resolution of inflammation. So this is a very big, quick summary of what we know about adaptive immune responses, T cells in atherosclerosis. But the idea here is to know whether this adaptive immune pathways can be harnessed for the treatment of patients. So this is in mice, but we know also in human beings. For example, my group focused on the role of Tregs and worked on how we can use these Tregs in order to potentially limit inflammation in vivo. So you can see that Tregs are important also in human beings. This is an example of Treg levels in patients, in control patients, in patients with stable angina, or in patients with non-ST elevation MI, where you have a reduction in the number of Tregs, but there is also a reduction in their function. There are other studies that have related and linked levels of Tregs in the blood to the occurrence of myocardial infarctions in large cohorts, as this work from Jan Nilsson's group. And based on this idea, we thought that there might be a way to modulate Tregs in vivo in patients and see whether we can reduce inflammation. And in fact, the way that we did this is based on this concept and the role of IL-2, interleukin-2, in T cell biology. What we know is that interleukin-2 at high doses activate effector T cells, here in red. So you take high doses of, thousands of IL-2 units per mole in order to activate STAT5 phosphorylation, which is downstream of the IL-2 receptor. And in fact, based on these data, IL-2 has been used and approved for the treatment of metastatic renal cancer. The idea behind this that was high dose of interleukin-2 will activate effector T cells that will activate T cells that will kill tumor cells. And it has been trialed and tested, approved for the treatment of metastatic renal cancer. The problem is that they've seen that some of IL-2 was also activating Tregs. In fact, if you look at this, ultra low doses of IL-2, so 1,000 times lower than the doses required to activate effectors, are enough to activate Tregs. And this can be explained by the fact that Tregs express what we call the high affinity IL-2 receptor, the trimeric receptor that combines the alpha, beta, and gamma chains. And in fact, this high affinity trimeric receptor is exclusively very highly expressed on Tregs, and very low doses are sufficient in order to activate this receptor and promote Treg expansion and activation in comparison with effectors. So people have tried this in autoimmune-mediated diseases, and we wanted to see whether low doses of interleukin-2 can be used in patients with cardiovascular disease. So we did a first study. IL-2 is contraindicated in patients with cardiovascular disease. So how do you start by testing this if it's contraindicated? So you go from scratch, and then you do a safety trial, and then you test different doses of IL-2 sequentially, in a sequential manner, in a stepwise increasing manner. So in this study, which is called LILACs, we started by patients with stable angina and tested 5 different doses of IL-2 in these patients to see whether they are safe and whether they are able to increase Tregs without increasing effector T cells. And so this is the dose range that we used, from 0.3 to 3 million international units. And we have seen in these patients that there is quite like a dose-dependent increase in Tregs within these range of doses without any increase in T effectors. Now, from this, we selected, mathematically speaking, when we modeled this, we selected 2 doses to be tested in patients with acute coronary syndromes. And we tested the 1.5 and 2.5 million dose. And again, we saw that these two doses were able to increase Tregs in a selective manner without increasing T effectors. However, we did, I don't know whether I have this here now, but we did a lot of mechanistic studies, with single-cell sequencing in order to understand, this is the first time, despite the fact that IL-2 has been trialed in autoimmune-mediated diseases, this was the first time in cardiovascular disease, and this was the first time that people were looking at what IL-2 is doing to the immune landscape in vivo in patients. So using single-cell studies, we determined that the 1.5 dose was the most appropriate dose in vivo, that was the most selective for Tregs. So we went on to the next phase with the phase 2b trial, where we wanted to test this dose of IL-2 and look at its impact on on vascular inflammation in patients. Because vascular inflammation and we looked at vascular inflammation measured by FDG PET-CT, because this is a known a good surrogate biomarker of cardiovascular events in these patients. So we randomized patients with acute myocardial infarction and high sensitivity CRP levels to placebo versus IL-2 and treated them for 8 weeks and did FDG PET CT imaging for vascular inflammation before and after treatment. And this is the results. This is the primary endpoint of reduction of vascular inflammation in these patients, and it was quite significant. So low-dose IL-2 reduces vascular inflammation in these patients in a significant manner. And you can look at different ways. This is the active segments, the highly inflammatory segments at the beginning, and low-dose IL-2 reduces inflammation in these segments. And in fact, if you look at the impact of IL-2 over placebo on the reduction of vascular inflammation, you can see that this is the baseline level of inflammation, and you can see that the higher the inflammation baseline level, the bigger is the impact of IL-2 versus placebo on reducing this inflammation. Now, we had, of course, very few patients in this study, 13 in each group, but we have seen positive signals of, in terms of cardiovascular events. We've had 4 cardiovascular events within less than 2 years of follow-up, and all of these cardiovascular events occurred in the placebo group and none in the low-dose IL-2 group. Now we are doing additional studies, just a few slides to show you that we are also interested in B cells. When adaptive immune T cells are activated, they also give help to the B cell part of the humoral immune responses, but B cells also, we know that they play, my group and others have shown before that they play an important role in controlling atherosclerosis. We know that depletion of mature B cells reduces atherosclerosis in mice. And we have also published papers showing that B cells are also activated at the acute phase of myocardial infarction and orchestrate the inflammatory response post-MI. In particular, they produce chemokines that attract monocytes, and this is important in adverse cardiovascular remodeling. So what we are, and in fact, other groups have published that post-MI, these B cells, besides, our demonstration that they are involved in adverse cardiovascular remodeling, this group have shown that B cell activation post-MI leads to antibody production and IgG immune cell complexes, and this brings more inflammation to the atherosclerotic lesions and promotes atherosclerosis progression. So what we are testing now is taking patients with acute myocardial infarction and treating them, this is the idea, with rituximab to deplete B cells at the acute phase of myocardial infarction and look at the recovery of heart function and vascular inflammation. And this trial is ongoing currently, and it's a European multi-center trial. Patients with anterior STEMI are randomized to placebo and rituximab with the primary endpoint at 6 months of heart functions by CMR in these patients. I'll finish this. So my conclusions would be that the immune system is really a central driver of atherosclerotic cardiovascular disease, with both activation of both innate and adaptive immune responses. We think that targeting immune system will bring benefit to patients. The important thing to work on is what kind of inflammatory pathways to target in order to bring effective reduction of cardiovascular events without inducing side effects. You've seen that targeting innate immune pathways has given us proof of concept, but at the expense of increased risk of side effects. And we believe that more subtle modulation of the adaptive immune pathways, in particular through regulatory T cells, may bring effectiveness and while ensuring safety. These are, the work of my great collaborators in Cambridge, in Paris, and in particular, Tian Zhao and Ruchal Suranjan for their great work in translating these concepts from bench to bedside. Thank you very much for your attention.
Thank you very much. Fantastic lecture, fantastic work. Very impressive. Matthias, my colleague Matthias Friedrich, who is a imaging and wears many hats, but particularly MRI imager, world leader. Matthias, go ahead. I'll let you ask a question.
Yeah, no, thank you very much. It's very interesting, and thank you so much for sharing such a great insight view of all these pathways. So my question is, taking it one step back, all these mechanistic insights rightfully lead to the conclusion that the immune system is key, at least I believe that key in the development and progression of atherosclerosis. But to what extent are these specific, let's say, genuine causes, or are they just triggered by our lifestyle, by unhealthy choices regarding diet, stress, lack of sleep, etc.? So could it be that we have to think about, okay, how do these mechanistic insights lead us to primordial causes of atherosclerosis, which then work through the immune system?
Yeah, I showed, it has for sure, our lifestyle is important. In fact, we know from trials like, the Mediterranean diet trials, where investigators have shown an impact on cardiovascular events. There are other studies from, sub-studies from these trials showing that this is also, associated, but these are association with also reduction in systemic inflammation. But I think that we should, again, be careful but when we talk about association. It doesn't mean that they are causally related. However, when I talk about LDL cholesterol, and I wanted to show you the intimate, intricate relationship between some of the most important cardiovascular, atherosclerotic cardiovascular risk factors and inflammation, we know for sure, from many basic experiments, preclinical models, that these factors, these causal factors of atherosclerosis, induce atherosclerosis
through inflammation.
Through inflammation.
Exactly.
I mean, these mice, have very high LDL, VLDL or LDL cholesterol levels. If they don't have monocytes, they do not develop atherosclerosis. So it doesn't mean that it is doing everything. And when we talk about monocytes, in fact, again, you cannot deplete monocytes in human beings, in order to limit atherosclerosis. And this is why we need to understand in more details what are the most important pathways that can be targeted and at what time point in life, because I'm pretty sure that not the same pathways act similarly and are of similar importance during different periods, in life. And this is very important. What we are doing now is that we are taking, and I was talking to Nara before, is that we are talking, analyzing atherosclerotic plaques from early on, very young individuals, children, kids, to people who are 80 years old, in order to understand whether at the cellular level and the molecular level, at single cell resolution, what is different between these plaques.
Yeah. No, I don't get me wrong. I absolutely, I also tell my patients, look, your cholesterol is less important than your lifestyle, because inflammation is what drives your, but that's exactly that's where they're related, because cholesterol also to me is not a cause for atherosclerosis. It's just a mediator, because the oxidized form of LDL cholesterol then can lead to intraplaque inflammation. No, my point was just, there's an opportunity, I think, to go from these mechanistic pathways to primordial causes, which is a lifestyle, pollution, whatever toxins we have, and stress, I think, in our society. So that was actually my point. But I think we're not too far away. It's just that I thought we could use that to extend it to get to the primordial causes. Thank you.
Thank you, Matthias. I have a few hands up. I have Stella. Stella, go ahead.
At my Lucian lecture, I will talk about some aspects of primordial prevention. So.
Hello, thank you very much for the presentation. Can you please comment on the interplay between the immune system, adaptive and innate, with non-immune cells in the atherosclerotic plaques in the context of stability and instability?
Yeah, sure. This is great because we think about the immune system as only within immune cells, but in fact it has a major impact on other non-immune cells, in particular the stromal cells. I'm not even talking about macrophages, because when I'm talking about IL-10, for example, produced by Tregs, IL-10 is a major anti-inflammatory mediator that can act on macrophages, but these are still immune cells. Now, about non-immune cells, there is and this is why I see the importance of regulatory T cells, because as I was saying this morning, I think that they go beyond just inflammation resolution. Tregs also can work as factory for growth factors, and growth factors that are important for tissue repair. Just amphiregulin, for example, this morning talked about skeletal muscle. It's very important in regeneration of skeletal muscle, but it's also important in regeneration of cardiac regeneration. It is important in anti-inflammatory mechanisms.
In
Now,
particular, vascular smooth muscle cells, endothelial cells.
yeah, looking at smooth muscle cells, for example. So we have a study that is coming that we are interested in. We randomized patients who are admitted for stroke to low-dose interleukin-2 versus placebo and took their carotid endarterectomy specimens to look at what IL-2 is doing inside the plaques, right? And in fact, one of the most important changes that we see initially is the accumulation of foam-like smooth muscle cells. So you can imagine that these Tregs are acting in a very different manner, and broadly, not only on the immune cells, but also on non-inflammatory stromal cells, and their action on the endothelial cells, smooth muscle cells, fibroblast has been described in the literature. And I think that this is one of the way that they can be important in tissue healing responses.
Dr. Stella, and then Dr. Kaberi, and then Dr. Kantor.
So I'm a physician epidemiologist, and I second Matthias's questions about health behavior, because that's my area of work. But my question to you is, could it be, it was really interesting how you showed the immunogenicity of LDL sort of triggering the inflammatory responses. Rather than going after different aspects of the inflammasome, would there be a way to make the LDL less immunogenic? Are there small molecules to sort of prevent it from triggering that cascade rather than cutting off the cascade, which has other beneficial effects?
So we know that limiting oxidation is not working, right? So, I mean, there was a lot of work on antioxidants in atherosclerosis.
There's no...
All trials failed.
Yeah.
Probably because, there are a lot of explanations. Some of the vitamins that were given have also pro-oxidant effects. Some of them, it might be also because there's no good tissue delivery, and but it doesn't the inflammation and the oxidation hypothesis is still important. Is that just how whether you are able to modify it in vivo or not? Now, one way of limiting the endogenous activation of the innate immune system or the adaptive immune system by modified LDL is also, to give to people, and people have tried this at least in preclinical models, is to develop antibodies against oxidation-specific epitopes that are expressed on endothelial cells. Work from Joe Whitstone lab showed that overexpression of antibodies against oxidation-specific epitopes, at least in mice, can limit the inflammatory mechanisms of these LDL and limit atherosclerosis. In fact, there is currently a trial by a company called Abcentra. They are testing in humans, in patients with acute coronary syndromes. They have an antibody directed against oxidized LDL moieties, and it is in trial. So we will know. So this is an additional other pathway that is potentially interesting.
Dr. Kantis?
Yeah, thanks, I have some questions about IL-6 specifically. So you mentioned that CRP is mainly an IL-6 readout. And so the question is, how important is IL-6? There's so many other cells that you show that are important. Is there a better marker for inflammation than just CRP? Are we measuring the wrong thing, or is it very limited in scope? The second question relates to the increase in IL-6 after exercise, which is supposed to be good for us. So I wonder whether we should be exercising only late or not going in the phase of disease.
Yes.
Questions, please, so we can.
Yes. So the first question is about IL-6 and whether CRP. When I said that CRP is dependent exclusively on IL-6 and whether CRP measurements is the right measurements as a readout of inflammation, I think again it's a readout of IL-6 pathway. Again, I can go ahead and as I mentioned, there are mutations in human beings of the IL-6 receptor pathways. These patients have zero CRP levels, despite the fact that they can mount other inflammatory responses. In humans, the genetic variant in the IL-6, and the CRP is downstream of the membrane signaling of the IL-6 receptor. So this is in the liver. Now, IL-6 can signal through additional signaling pathways called the trans-signaling pathways in other cell types. And this requires IL-6 with its soluble IL-6 receptor, and it can signal in any other cells that have the GB130. Again, this pathway, you don't know what the trans-signaling pathway is doing in atherosclerosis. So this is why I cannot tell whether the IL-6 inhibitors will be reducing cardiovascular events, because they don't reproduce what the genetic variant does, right? So it's open. It's still open. The other question is, when we exercise, there is an increase in IL-6, which is true, and it's supposed to be detrimental in atherosclerosis. So why exercise is good? Because this increase during exercise is not chronic. Is from time to time, right? You increase if you have very intensive exercise. There is an increase in IL-6, and in fact, IL-6 act on stromal cells. It's a very good growth factor and reparative factor. You know, all of these is that how you interpret this in the long term. And exercise is not only IL-6, you know. It's only IL-6 is only part of the cytokines, mediators, growth factor, chemokines that are produced during exercise. So it's very difficult to say, you know, you cannot say because exercising increases IL-6, then it's not good. In fact, we know that exercise is one of the most interesting treatments, you know, for cardiovascular disease. You know, you exercise, and this is the most highly effective drug, let's say.
Sasha, did you want to join the conversation? You have some comments in the chat.
So, yeah, so thank you again, and Matthias and Kaberi brought up the same thing, which was these lifestyle factors that so my take on this theme would be, if you give rituximab to 100 patients, that would cost our facility $2 million. And advising somebody to eat a plant-based diet or something similar, exercise more, costs almost nothing. So, but I see the value, just like Matthias and others, we see the value of this kind of research because it gives us an understanding of what's going on. So I wonder how you feel about translating this. Do you like? Do we? And here's the other thing: like the Choosing Wisely program in Canada. One of the things we feel we should be doing now as physicians is deprescribing. So if we think now we have to add another drug, a small molecule or a biologic, to cardiovascular patients who are already taking a number of other therapies. So would you see, in your knowledge translation of your work, could you see this leading to not necessarily a drug, but maybe ammunition to explain to the patient why it is important to eat a heart smart diet or lose weight or whatever?
I'm a big advocate of primordial and primary prevention. Don't misunderstand me. What you are doing here is at very advanced stages of the disease setting. Okay, these patients still need treatment, right? So we need to find treatments for these patients because they are still needing it. I started a bit late maybe, but personally, I don't have any elevation of LDL cholesterol. Usually my LDL cholesterol, I don't know, you think about milligram per deciliter here?
Millimoles per liter.
So millimoles per liter, I think mine was around 2.5, which isn't very high. And I'm under rosuvastatin 20 milligrams and ezetimibe 10 milligrams, myself, I don't have any other risk factors because I believe in the fact that you have to have your LDL cholesterol the lower, the earlier, and the longer. So if you can start by lifestyle measures early in life in order to, we are born with around 70 milligrams, 60 milligrams per deciliter, around 1.76 millimole per liter. And during the first years of life until 18 years, it goes up to around 110, 120. And this increase might be, mostly related to lifestyle, but there is a component genetic part that you cannot do anything about it, right? But there is another additional lifestyle part that you can do something about it. And I'm a big advocate of, starting, the earlier the possible and the longer the possible. But I'm talking here about something that comes later. And in terms of inflammation, in terms of primordial prevention or primary prevention with inflammation, acting on the inflammatory system, the immune system, this in my mind could come with vaccination. And we are working on this. You can modulate and manipulate the immune system in order to instruct it to react by promoting Treg responses against these oxidation-specific epitopes and reduce plaque inflammation and plaque development. And this could come by some kinds of vaccination, which we take to prevent viral infections and others, right? And this is the way that we can think about later on, when we have identified the specific, antigens that are the most interesting, the specific TCRs, the modalities of inducing good tolerization. These are potential ways of limiting the activation of the immune system very early in life.
Okay, fantastic. Wow, thank you. Thanks everyone for joining the discussion, and thank you for this wonderful presentation and sharing your thoughts and with us. For those of you who are interested, there are more lectures coming as part of the Lucian Award. Sylvia, I will ask you to please, she will email everybody who's on the list when the next lecture is and how to listen to it, whether in person or virtually. Thanks everyone for joining us, and thanks again for the fantastic lecture, and welcome to Miguel.